yourself. Okay, Peter, please, I give the time to you. And would you like, please introduce yourself a little bit, including your CV, because I'm so sorry, I have no CV at all for you. Okay, time is yours, Peter.
Okay, so thanks a lot for the friendly introduction. Just give me a second. I have to get the presentation right. And maybe can you give me the presentation right so I can show
Yes,
my slides?
I mean you are now is as a co-host, I would say, and you can share by your own because you are now co-host. Let me check, Peter.
No, not working yet.
Oh, wait a minute. You are a co-host already, I would say. Edo, could you just help me? Can you see that Peter also co-host? Yes, you are the co-host, Peter.
Ah,
And
yeah, now, okay.
okay, you can share your PPT.
Okay. So I hope you can see it. Can you see the slides?
Yes.
Perfect. Okay, great. So, selamat pagi, everyone.
Selamat
Nice to
pagi.
have you here. It's the little Indonesian I learned during my time at
Wow,
university
perfect.
together with Arif. Yes, I will give you a short introduction about microbiology in the pharmaceutical industry. I will focus a little bit on good manufacturing practice at first, dive a little bit into biopharmaceuticals, what one might do as microbiologist in the pharmaceutical industry, and then we will head on a little bit. So in case you have any questions, just ask. And I will show you or tell you a little bit about myself. But before, I have a little disclaimer. So the opinion expressed in this presentation are those of the author, so of me, and they do not purport to reflect opinions or views of Boehringer Ingelheim, so the corporation where I'm working at, its members or other parties, and the information presented within does not necessarily reflect the corporate opinion. The presentation was created for education purpose only and falls within the standards of fair use. So, okay, but that's it. So, that's me. I started my academic career in 2007. I studied biology at Eberhard Karls University Tübingen in Germany. And following my biology studies, I followed up with microbiology until 2013. And from 2013 to the late end of 2016, I did my PhD, or in Germany, it's called Doktoranat, on the topic New Insights into Antimicrobial Peptide Resistance of Staphylococcus aureus. So basically, I spent my whole academic life researching Staphylococcus aureus. And I will just show you some pictures of Tübingen. So it's a really, really neat German town, a university town. You have this beautiful river, Neckar River, the old university. And we have this nice castle Tübingen. So it's really a great place to study. And this was my professor. So also the professor of Arif Luckmann, Professor Friedrich Götz. He's
I met him twice.
Really? Okay.
Yes.
He's really a nice guy.
Yes, true. That's true.
He's an excellent Staphylococcus researcher. So he really pioneered the field of Staphylococci. And so this is the University for the Natural Sciences in Tübingen. Unfortunately, it's not that pretty as the old town, so it's rather new, lots of stone and so on. And they're also expanding a lot. They're building new biochemistry departments, plant physiology departments. So Tübingen is really, if you have the chance to go to Tübingen, I could really recommend, or I would really recommend.
I love it.
Yeah, it's really a nice town. Good place to study. And just 2 snapshots from the lab. Not very tidy, as you can see, but that's where we work, where we did our PhD. And you can also see Arif when he was not.
Arif correct?
That's Arif, correct. Yeah, when he was not PhD yet. So really, we spent a lot of time together in the lab with some more students, and yeah, it's like PhD was really good time. So lots of struggling, but also lots of fun. And yeah, at the beginning of 2018, so I decided to leave the academia and to join the industry. And now I'm at Boehringer Ingelheim since April 2018 as trainee. So I started as trainee in pharmaceutical production, which basically means that you go into different departments, you see manufacturing, you see quality assurance, quality control, and so on. And since January 2019, I'm a compliance officer and deputy head of production at facility Mucosolvan, also in Biberach, Germany. Yeah, that's what my current profession is. And I will also tell you a little bit more about that. But at first, some informations about Boehringer Ingelheim. Sorry, wrong direction. So Boehringer Ingelheim is one of the world's 20 leading pharmaceutical companies. So it's really a large company with like 50,000 employees worldwide. It was founded in Ingelheim in 1885. And what's really outstanding that it's still a family-owned company. So it's not listed on the stock market or so, but the family still controls it. And Boehringer has more or less three divisions where it's focused on. So it's the human pharma segment, animal health, also with vaccines, and biopharmaceutical contract manufacturing. So Boehringer Ingelheim also manufactures biopharmaceuticals like antibodies for other companies. And another thing that really stands out about Boehringer is that it focuses a lot on research and development. So it spends roughly 22% of it human pharma net sales on research and development. So we have lots of own products in our pipeline, and that also adds up to this light build or to this slogan, value through innovation. That's very important for Boehringer. So the key indications in which Boehringer is active is respiratory disease, immunology, and oncology. So oncology therapy is really hitting up a lot. So that's where a lot of stuff is going on. And like I already said, Boehringer does this biopharmaceutical contract manufacturing. We have production plants in Germany, in Austria, and the US, and also in China. And Boehringer is the social responsibility, which stems a little bit from this family-owned company. So they really they have this Making More Health initiative, where they really focus on improving health in other countries, in low-income countries, helping social or doing social projects. And it's so there are more than 6,000 employees on the site in Biberach, and it's the main research location for BI. So most of the development and research is going on here. And it's also the largest European production plant for biopharmaceuticals. And as you can see in this Google Maps picture on the right, so Biberach has around 30,000 inhabitants, and 1/5 basically is employed at Boehringer. So okay, let's head a little bit further. So speaking of biopharmaceuticals, which are produced here, I think what you all know are peptides, and now what's really getting more and more important in the pharmaceutical industry are biopharmaceuticals, for example, proteins, or like in this case, antibodies, which are used for treating, for example, cancer. And what you can see is that there's a huge increase in sales during the last 15 years. So maybe until 27 new chemical entities, so like these, like small chemical structures, were very important in the market. And until 2018, biopharmaceuticals like recombinant proteins, antibodies, cultured cells, blood products, and so on are really hitting up the market. It's an incredible growth of these products. And no wonder in 2019, the best-selling drug on the market, or medicine on the market, was Humira, with roughly 20 billion US dollars in sales, followed by $12 billion sales of Elquis, which is a NCE. So biopharmaceuticals are really, really important. They will take more and more market share. So it's not that the chemical structure time is over, but it's really that biopharmaceuticals will gain more and more momentum on the market. So just to give you a rough idea about pharmaceutical production, I guess that's it's actually not so different from what you can do at a university in the lab. It's just on a way, way bigger scale. So usually you will start with your stock culture, which most of the times are Chinese hamster ovary cells, but there are also some microbials around. Put them, or you grow them in the flask, prepare the inoculum, and then put them in the bioreactor for production. Let them grow. The proteins or the antibodies are produced. Cells are centrifuged, filtrated, and the proteins or antibodies are basically extracted. There are a lot of chromatography steps, polishing, viral filtration, and when you have then your antibody, it goes to formulations, basically where protein, buffer, salt, and protectants are added. It's dry freezed, undergoes quality control and packaging, and basically you have your final biopharmaceutical. So it's not so different from what you would do at the lab when you're purifying antibodies or your recombinant proteins. It's just like a way, way bigger scale. I would say the biggest difference to the lab is that most biopharmaceuticals are still parenteral alia. So you would inject these antibodies or these medicines, and for lab research, it's not, I would say it's maybe not so important that it's 100% pure. It's at least, or as long as it works in your assay. But if you inject it into humans, of course, drugs should be of high quality, they should be safe to use, and they should also be effective. I think that's clear. Which brings us to GMP. GMP is basically the framework in which the pharma companies are producing the products. So it stands for Good Manufacturing Practice. I'm not sure how much knowledge you all have, so we'll just elaborate a little bit on it. At least in the German university, you hear of it, but you don't really get to know what it means. So Good Manufacturing Practice is a framework to ensure that products are produced and controlled to define quality standards. And these quality standards are basically defined in relation to the intended use of the drug and the regulatory environment, which basically just means, like I also said, if you have medicines which are injected, the standards might be way, way higher than if they're just orally taken, like capsules or tablets. And it also depends on which market you go. So, for example, US and Russia might have higher standards than other countries. And that's basically what it's about. It's a highly regulated environment in which we are working. So GMP basically comprises drug production, so how the drugs are produced, but also how media are used for production. For example, water, but also air or nitrogen are produced, how our APIs are produced, so our active pharmaceutical ingredient, so basically the pharmaceutical active compound like aspirin or the antibody. GMP is involved in the testing of drugs, so the quality check basically, packaging of drugs, so that they're sealed correctly, they're stable, they won't get bad on the way to the customer, and also for market release of batches. So there's a lot of documentation going on. Every production step has to be checked before the batch actually hits the market. And yeah, this whole regulatory thing is not only confined to the manufacturing, but also to other steps. So it's not only GMP this time, but also GXP. So we have GLP, GCP, GSP, GDP, GACP, GEP, and so on. So lots of acronyms, which basically means all the steps after production or during production and on the sidelines are controlled by regulatory yeah, or document, yeah, regulatory documents and so on. So basically, all the research you're doing has to be regulated. All your clinical studies have to be regulated. How you store stuff is regulated. How goods are distributed is regulated. For example, you wouldn't want your medicine to be in the truck for too long, to be exposed to high temperatures because they might get bad, and so on and so forth. And this basically hits the whole side production lines of biopharmaceuticals and other pharmaceuticals these days. And this is like not a European or US thing, but basically manufacturing of drugs is on an international base because they also market internationally. And there are a lot of legal bases and influences. For example, these are the one which are on the left side. These are the one which are most important, at least for Boehringer, the EU GMP guideline and the German legislation, but also the US 21 CFR and the ICH guidelines, which is very important for us, and the World Health Organization technical report series. And I'm I'm not a specialist for this because we don't have I don't think we have so many products on the Indonesian market, but there's also this National Agency for Drug and Food Control, which might add up to the quality management systems. But I guess there are people who can explain that better than I could. Yeah, so what I point out is basically that we have all these international guidelines and laws and so on and so forth, and they all are going down or manifesting itself in our quality management systems. So the quality management systems basically define how we work, how we manufacture, how we control our product. And these are some points which are present in this quality management system. So we have change control. Basically, if we change anything on our production machinery, it has to be documented and controlled. All our machines have to be qualified and validated. We have standard operating procedures, which basically exactly tell you what to do and how to do it. We have supplier management so that we have trusted suppliers, risk management, complaint management. So it might be that your product on the market has some defects or you have some side effects. That's all followed up. We have self-inspections and inspection by regulatory authorities. So basically, inspectors come to our plant and say, okay, we want to test or please show us that all your production is going well and everything is fine, and so on and so forth. So it's just like, it's a huge, bulky, regulated field in which we are working. And that basically makes pharma companies really, really big. You need a lot of people, not a lot of departments. Yeah, okay, so I hope that's for the more or less boring stuff. We are here to talk a little bit about microbiology in the pharmaceutical industry. So the question is, as a microbiologist, what could you actually do when working in the pharmaceutical industry? So like I already elaborated a little bit on biopharmaceutical production. Basically, that's one thing where you could get a job, for example, as process advisor or fermentation specialist. So if you're really into fermentation, that might be a good start. Coming from the university, of course, research and development makes sense. For example, a scientist, you can pursue postdoc career or even become head of laboratory. Quality control and in-process controls are very important. So basically, if you're good with HPLC and so on, that might be a field. Or if you're more interested in medicine and clinical research, you could aim to become medical science liaison officer or clinical research organization monitor. Or I would say the biggest part, actually, where the most jobs are available, are the quality departments, so quality assurance. For example, as quality officer. So these are the guys that basically all the guidelines given by these international laws and so on are followed in production. And yeah, like I told you, I'm a compliance officer, and the question is, what am I doing? So I'm a little bit in between those worlds. So I also sit in the clean room. So I consider myself, or my department consider itself, to be part of the production, but we also have, more or less quality aspects on our working schedule. So I would say our main task is basically to secure manufacturing along guidelines and SOPs, so that we get a good and safe product, but also to support. Yeah.
Yeah, I'm so sorry to disturb a little bit. I would like to inform you. Because now at my home home area is very heavy raining, and also the water is now increasing since maybe water will go to my home. I don't know. This is so big. That's why I'm leaving this meeting room. And Ibu Budaya, are you there, Budaya?
Yes, Budaya.
Okay. Peter. Yes, thank you, Budaya. Thank you so much for helping us.
Okay, Bu Maya.
Budaya will be as the moderator for the next. She will be there as the moderator.
Okay, no problem, Bu Maya.
Okay.
Sorry, I should meeting because the water is really coming. Okay.
Okay, you better check your house.
God bless you. God bless you.
Bye.
Bye.
The other of you, can you hear me? So is the connection, is it good, or am I too fast? Is it fine?
Yes. No, fine.
Okay, great. Okay. Because it was hard to understand her right now a little bit for the connection. Yes. But if you can hear me, it's okay. And please tell me if I'm too fast or if the connection gets bad. It's no problem.
Okay.
Okay. So as I said, there's just different activities I have in my working routine. So also to support the production process. So I have some technical knowledge about all the machines we are using. Very important is housekeeping. So to ensure that all the facilities are clean and disinfected, but also qualification and validation of equipment. But also, change control is part of my work, deviations and OX investigations. So basically, when we have incidents, a machine is not working correctly, I have to, basically judge whether the product is still good or not. I'm responsible for training of the staff, but also for communication with in-house services, service providers like logistics, janitors, cleaning staff, and I have projects with other business units within the same production facility. So this compliance officer, for me, it's quite an interesting job, because you're a little bit in between those worlds and you get to know a little bit about everything, basically. Unfortunately, coming back to microbiology, me as a microbiologist, so there's not so much actual microbiology in my daily routine. So I highlighted these points where microbiology plays a role, and mostly in the parts of hygiene. So unfortunately, I'm not doing any fancy research or development projects with bacteria, but it's more that I really have to control that our hygiene status is good. There are no pathogens in our production. The bacteria levels are fine of the product, and so on. So I will go a little bit deeper into that. So yeah, bacteria and pharmaceutical production. Yeah, I think everyone who's working in the lab knows that bacteria can actually produce the active pharmaceutical ingredient, for example. So bacteria can be used for production of biopharmaceuticals. Also, most of the time, especially concerning antibodies, you would rather use yeah, eukaryotic cell lines. Bacteria can also be used for production of excipients, or, for example, as drug itself, so microbiome treatment, which gets more and more important, so like Lactococcus lactis, for example. But to be honest, the biggest thing, at least for us, is bacteria as contamination of our drug. So basically, you can see the cell lines during the fermentation process, and these are definitely contaminated with bacteria, which is not what we want. So you can basically, or just to make it a little bit easier to understand, maybe you can divide it, the whole manufacturing process, into two parts. So production of the drug substance, or the API, which is basically the fermentation. And as you all know it from the lab, if you grow your cells or your bacteria, you always have the possibility that you have a contamination of the culture broth, and you won't get the protein or the plasmid or whatever you're purifying as you like it. And the second part would be contamination of finished drug during fill and finish, so the drug product. So this is basically when we already have our antibody or biopharmaceutical, and it's formulated, lyophilized, and filled into vials. And basically in this picture, you can really see you have this turbid growth of bacteria in the vial. So that's definitely nothing that you would want to get injected into your body. And these are the things basically which play a role. So reasons might be, for example, within the production process, that we have faulty process, or our barrier systems are broken, so that basically bacteria get inside of the fermenters. There might be contamination by intervention. So for example, sterilization of the process equipment was not effective. So we have non-sterile equipment which we are using, or which is a big factor actually, is contamination through human stuff, faulty personal disinfection. It's basically in biopharmaceutical production, every step which is undertaken by human staff is yeah, a potential contamination danger. So yeah, and so how to prevent these contaminations? And one big part is hygiene, actually. So which on the one hand boils down to production hygiene and staff hygiene. So like on the cartoon, as you can see, these production guys, so they don't really look very yeah, they have dirty clothes, their hair is outside of the protective nets and so on. So that's definitely something you wouldn't want. And the second thing is that you have robust production processes, so that you don't run in danger of any contamination of your product. And I will elaborate or go a little bit deeper into this production hygiene, staff hygiene aspect, and basically how we deal with it in the production. So yeah, like I said, a hygienically clean environment is one of the key requirements for production of high quality drugs. And if you think about producing these drugs, it's obvious that you cannot do it like in the lab, but there are lots of things to consider. For example, size and positioning of your rooms and your machines, design and positioning, disinfection of your drains and outflows in the production facilities, disposal, how to get rid of your waste, how to organize the storage of it, sanitary facilities. Of course, your production workers at one point have to go to the toilet, drink some water, and so on. So where to put those? Are your cleaning and disinfection agents actually active? Can they remove the bacteria? So they have to be qualified. and yeah, social rooms, where to position those so that you don't have too much traffic in your production facilities. So these are some points. And yeah, like I said, like on the left side, steelwork or weird car factory or car workshop, it's definitely not how we can produce, but all the production is going on in clean rooms. So clean rooms basically are an environment that has a very low level of environmental pollutants, such as dust, but also airborne microbes, aerosol particles, and vapors. And it has a controlled and very important documented level of contamination. So documentation is very, very important for us. Coming to sources of contamination within the production, of course, like I told you, rooms or machines might be sources of contamination, but also packaging materials. So everything or all the starting materials which come into the facility, they might be contaminated, so they have to be checked. Process gases like nitrogen, oxygen, which basically directly are used in the process, have to be controlled. Tools, but also the ambient air. So as you can imagine, in a clean room, you just cannot pump the air from the city outside inside the room, because yeah, it would not be very good. So big part for this is that you have defined hygiene procedures for production rooms and equipment. So these procedures have to state which rooms and which equipment has to be cleaned, how often it has to be cleaned. Are there any extra cleaning activities because the production process maybe makes it important because you have lots of materials moving around in the room? Which cleaning and disinfectant agents are used? So for example, some production plants might have problems with spore forming bacteria. So you would need disinfectants which can really kill the spores. Some might have problems with fungi, so you need anti-fungal agents and so on. Is the state of the cleanliness visible? So can the production worker judge whether the room is disinfected or not, whether he can start to work or not? And like I said, documentation, because like one big rule, if it was not documented, it was not done. And this is especially important for inspections. When the inspectors of, for example, the Federal Drug Administration or so come, they will check your cleaning schedules, and yeah, so you have to prove basically that all the cleaning was done and your rooms are clean, and you can produce a proper product. Yeah, this makes our framework very complicated, actually, because this definition of hygiene procedures is all manifested in these standard operating procedures. So for example, in our case, we have one general cleaning standard operating procedure, which gives you the rough framework or the rationale how often the rooms have to be cleaned, which materials are inside of the room, of what materials, for example, doors are made of, and so on and so forth. And this document splits in further documents, which are defining, for example, the hold times. So how long after cleaning an equipment you can basically use it, when do you have to clean it again, and how long are you allowed to keep it uncleaned. So these are all things. Allowed cleaning agents. We have an own document for allowed cleaning agents, for allowed cleaning tools. So for example, we're not allowed to use brushes which are made out of what is it called, hair from animals. So you only are used to you're only allowed to use synthetic brushes. The laundry programs are defined, and our whole cleaning concept is basically defined. And for the cleaning concept, so that's just to give you an idea how it might look. Like every item, every part of the room, every equipment is defined in this cleaning concept. For example, walls, doors, and ceilings, when, weekly, and as required. What to use, disinfecting, cleaning, with what, so the respective cleaning agent, and who does it. And this is very important. It's risk-based, and all these cleanings have to be documented. So cleaning and hygiene is a really big part of our daily life. Another thing is basically how your production facility is designed. I hope you can see that. It's maybe a little bit small. So basically we have a plan with a defined and documented flow of materials and personnel. So it's basically defined how personnel and material is moved within the facility, within the rooms, to prevent cross-contamination due to overlapping flows. And it's also risk-based. So basically, if you build a new production facility, you have to provide a rationale why some things are done like that. So in this example, basically, you can see the gray arrows. This is basically the way of the raw material. It enters from the left from our logistic axis, goes down to the south into the production storage, and from the production storage, it goes further into the first production rooms, for example, container filling. Here we still have paper bags and so on. And from these rooms on the north and on the right side, no more paper is allowed. They're put in aluminum boxes or steel boxes and so on. So this is really an important thing so that you don't get too much dirt or bacteria from the outside into your plant. Yeah, talking about starting materials and packaging materials, we have incoming goods inspections. So the laboratory basically looks at the microbial count. They take samples depending on the product and check it. We have to repack. That is what I told you. No card boxes or paper drums allowed. So basically, we repack all our starting materials into suitable containers, like aluminum boxes and so on. And we have cleaning procedures before entering of the production area. So basically, if the materials are transported into the production area, we have to clean the wheels of the wagons. We reload materials. If they're delivered on wood pallets, they have to go on alloy pallets and so on and so forth. So it's just to really keep sure that no outside bacteria, germs, or dust is coming into the production rooms. And the same, or we have also barrier measures in case of process gases. So for example, if we need nitrogen and oxygen for our process, we have sterile filters installed so that in case of contamination, nothing gets into the product. And these gases are also monitored if they have contact to the product for microbial count, but also for particles, for water, and for oil content. Yeah, that's very important so that no oil or water gets into your product, which you really wouldn't want there. And same true for the ambient air. So before the air enters your production room, it's preconditioned, so it's filtered. We have a defined temperature and humidity. It's deionized, and we have a laminar flow and pressure and flow control. It's in German, but I think you get the idea basically. So we have two airlocks on the left, one for material and one for personnel. And then you have the production room with the HVAC system, so basically with the ambient air going into the, what is it called in English? Oh, I'm not sure. Yeah, basically the ambient air is provided for the production room by filters and so on. And important is that basically the pressure is higher in the production rooms so that no contamination from the floors or the hallways gets into the production room. So you saw the picture of the, or maybe I switch back. You saw the picture of the worker dressing himself. So they have to put on these clean room gowning. And to support these gowning procedures, they are also defined. And the airlocks are designed to basically support this process. And maybe, I don't know whether you know the word pokayoke. Pokayoke basically means that you design a process that way that it's harder to fail. Like, for example, if you go to the ATM and withdraw cash, usually you will get your bank card back before you get your money, so that you take your bank card and you will not leave it there. And that's what we also have in these gowning or rooms or airlocks. So you enter the room, you put your things into the drawer, you put on the first pair of shoes, you wash your hands, you go further into the airlock, you put on your hairnet, you put on your beard net if you have one. So that's important for women, of course. And you put on your clean room suit, you put on your clean room socks, then you enter, you go on the bench and put on your production shoes, you disinfect your hands, and then you're allowed to go to the production area. And with this, we really try to keep all the dust and all the basically dirt we don't want to have in the production rooms outside. So lots and lots of organizational principles we have in the production. It works very well for us. We say, okay, we have a good production facility, but when the inspectors from the health ministry or from the FDA and so on, they come, you have to prove it. And one thing is, of course, to take them into the production and to see how it all works, but they also want to see documentation. And they would ask you, okay, that's all neat and fine, but how do you prove that actually hygiene status of the facility is fine, and how you track and how you prove it? And the only thing to do this is monitoring. So we're doing a lot of monitoring in our facilities. It's put under the roof of environmental monitoring. So we basically have monitoring of particles, so how many dust particles are within the room. We have monitoring of our pressure, so that we have positive pressure in the rooms, that no dirt from the outside gets sucked into the rooms. We have monitoring of our air exchange rate, so that the air is not standing too long in the rooms. And we have temperature and relative humidity monitoring for one part, because the product might get damaged if the humidity is too high, or also if it gets too low or the temperature is too high, but also for the personnel, because with very, very low humidity or too high humidity, it's not so comfortable. We have microbiological monitoring for air, basically for the quality of environment. We have personal monitoring. So we take agar plates and put it on the personnel, on the gloves, on the gowns, on the nails, personal hygiene. And we have monitoring of the surfaces. So to see basically, are there any bacteria that we wouldn't want in our facility? And depending on your clean room level, you have basically different levels or different amounts of bacteria and particles which are okay to have. In our case, we are producing non-sterile medicine. Grade D is sufficient, so 200 CFU per cubic meter in the air sample are fine for us. But as you could imagine, if you are filling sterile biopharmaceuticals which get injected, you wouldn't want any bacteria inside it. So basically the cleanliness or the levels which are acceptable increase from D to A level, while A being the highest level. And the same is also true for particles, so basically for dust particles and so on. If you're working with solid medicines, you have rather big limits, so it's fine. But if you go to the sterile manufacturing, you have very, very low limits, so only very little dust is allowed within your clean rooms, which makes sense, basically. Concerning this environmental monitoring, so the question is basically, okay, I told you a little bit about this environmental monitoring and so on and so forth, but how do you actually decide how often you would probe a room or probe an equipment? And this all has to be derived from a risk analysis. So depending on your room type, the risk of contamination is higher or lower. So for example, a production room is more critical than a storage room or an office room. So you would have to increase the monitoring frequency. The rate of cross-contamination plays a role, whether you have high, middle, or only low flow of staff and material. So basically rooms where you have lots of people going in and out, lots of material going in and out, and they're more likely to be contaminated, so you need higher monitoring. And ambient conditions, I think as you all know, bacteria like a humid, warm environment. So if you have a humid, warm environment, you need to monitor often versus dry environments. And also process conditions play a role. So basically dust formation and staph interventions are critical. And if you have no interventions, no dust formations, the risk of contamination is way, way lower. This is basically the framework of this risk analysis. And this is all calculated. So you assign numbers from 10 to 1, where 10 being the highest score, 1 being the lowest score. Then you add it up, basically room type times cross-contamination times ambient conditions times process conditions, which gives you the criticality. The higher the criticality, the worse. And the worse, the often you would have to monitor this room. And two more things that you would have to consider, or you can consider in this risk analysis, is that the criticality might be a little bit lowered if you have, for example, control mechanisms in place, like these ventilation, not valves, but these ventilation systems, which might suck off dust and so on during the production process. Or if you have a room which is only seldomly used, you would also not have to monitor it that often. So basically, criticality times control mechanisms times frequency of use gives you a risk priority number in your risk analysis. And this risk priority number basically is the basis for how often you would do a monitoring of your rooms. So, for example, storage rooms on the left, which where there are only few people coming in, you would have a low risk priority number. For example, only quarterly monitoring of particles and bacteria, while in production rooms where production is going on, but also not so many people are inside, and it's quite a good process. You maybe have only monthly monitoring. And in sculleries, cleaning kitchens, or sieve rooms where you have lots of humidity, lots of dust going on, you would need to do a weekly monitoring, for example, so that you can be really, really sure that at all time your dust and bacteria levels are at an appropriate level and there's no danger to your product. And yeah, how do we do that? So in our case, for example, every week we go through the facility, we follow our plan, which is derived from this risk assessment, and we do surface monitoring by placing these agar plates on surfaces and swabbing equipment, staff monitoring, so basically to see whether their gowning procedure was right and there are no bacteria left on the clean room clothing. We have these active airborne microbial count samplers, which basically suck in a defined amount of air, and they put it on an agar plate, and yeah, you can put these in the incubator then and count your airborne germs. Or we also have this passive settle plates, so which you just open, put in the production room, let it stand there for a defined amount of time, and then see whether you have some growth on it or not. And when we do this every week, we bring these plates to our microbiology department. So hopefully the plate will not look like this, because if the plate looks like this, it will mean that we have a bad hygiene status. And the microbiology department basically will count the bacteria growing on the plates. It will ID the bacteria, whether they are pathogens, whether they are human derived, which might be a hint that our gowning procedure is not so good, whether it's spore-forming bacteria or fungi. All these data is then stored in a database where we can evaluate it to do our trendings and reports, basically. So we do quarterly trendings where we can see, okay, our hygiene level is good or it's not so good. And in case we have action limit excursions, so if we have high bacteria number, or we have human-derived bacteria or spore formers, or negative trends, so basically increasing numbers of bacteria, we would have to activate our deviation system to investigate what's going on. Do we have a problem? Is our cleaning agent still active? Are the cleaning procedures followed? And so on and so forth. And basically to improve our hygiene concept. Okay. that's basically about it, about the hygiene part. As I told you, it's it's a little bit funny as microbiologist because it's not very fancy microbiology. It's yeah, it's daily work. So, but that's what I do, what I spend a lot of time with in my job. But I quickly want to give you some more information about the checks in the production process of biopharmaceuticals. Also, I have to say that I'm not an expert on this because in my facility, we are not manufacturing biopharmaceuticals, but solids and not sterile forms. And to go back to this robust production process, just to sum it up a little bit, basically important for the production of a robust production process is selection of starting materials, so that the starting materials you put into your product already have low microbial count levels. There are no pathogens or toxins inside. So that's why basically the starting materials are tested by our QC. During the production process, we have steps to reduce a pathogen or bacteria to ensure safe medicine. And this all goes down to, again, GMP. So by following good manufacturing practices, we are able to produce a high quality drug or medicine. And to come back to the fermentation process, so just as an example, on the left side, you would have your fermenter with your cells, and you can see these rod-shaped bacteria, which are basically small bioburden contamination. It depends on your product and where you want to distribute it, but some level of bioburden might be okay or might be within the regulations. So you might still be able to ship this product if there's very low bacterial or bioburden. However, as you know, bacteria also shed subcellular components, so cell wall or lipoproteins. And while bacteria are filtered out of the production process, or during purification with a sterile filter, 0.2 micron filter, subcellular bacterial components might still get on the column, might be co-purified with your antibody or with a biopharmaceutical, and at the end might get into your pool vessel. So you will not only have your product inside, but also leftovers of your bacteria, which you wouldn't want to have there. And so absence of the subcellular structures is very important for the quality of the drug. For example, protein exotoxins could be inside, like hemolysins, mycotoxins derived from fungi, flagellin, lipopeptides, or cell wall polysaccharides. And so these all would have an effect on the human or on the patient, especially if you think about cancer and they have already a suppressed immune system. This is very bad, of course. And you might also have proteases or other bacterial enzymes inside, which might lead to degradation of your drug. So maybe also your antibody is not followed correctly, or it's cut, and so on and so forth. So you really wouldn't want to have that in your product. And for this, there's a plethora of microbiology sub-laboratories in most companies. So for example, in Biberach, we have a water testing laboratory. We have bioburden testing. So they track the drug substance, so the fermentation process, whether we have contamination. We have sterility testing for the drug product. So basically for the filled vials, if they are okay. We have the lab which is evaluating our environmental monitoring. We have PCR and rapid methods laboratory, virus testing laboratory, endotoxin testing laboratory, and mycoplasma testing. So it's really a lot of laboratories. But also here, it's not like they're doing research or development. Most of the time, it's like very basic work. They have their assays and they run it from day to day. So it's lots of routine work going on. Okay, with this, I'm at the end of my talk. I hope I could give you some impressions about being a microbiologist in the pharmaceutical industry. So terima kasih, and greetings from Biberach, which you can see on the left. It's also a beautiful German town, and the local beavers, because that's basically where the name Biberach stems from, these animals, which I can actually see if I go by bike to the production facility to daily work. You can see them on the riverside sometimes. So if you have any question.
Okay, thank you so much, Dr. Peter. That was really awesome, your presentation and your explanations. Okay, you give us a really deep insight about the pharmaceutical industry, actually. Yeah, okay. I think we already have many questions here regarding the microorganism, especially in the pharmaceutical industry. We will making a bold underline here that hygienically clean environment is one of the key requirements for production of. Next, we will continue to the discussion sessions. Dr. Peter? Dr. Peter?
Yeah.
Yes, okay. I'm sorry. So
you can start. No,
I will I'm sorry. I will, yes, because yes, you can see in the chat room here, there already one question for say that for sterility test acceptance criteria in your company, is it still using a regulation that wait the result in around 14 days, or already using regulation by checking the documentation of the process only? How about the regulation
it's good, very good question. The problem is that I have not so much contact with the sterility testing. It's not really part of my job. I only know this by some colleagues. So don't nail me on that answer. But I don't think that we have it by only checking the documentation of the process. I think we still have sterility and testing. It might depend a little bit on the product, because it's also a part of the registration forms which you handed in. But I don't think we have this. We have the documentation, or we have the process only. I think we still have and sterilization, and sterility testing.
Okay. Okay, is it clear?
But as I said, I'm not a specialist for that. Sorry.
Okay. Maybe that's the answer. Wicatur, can you get it? Wicatur? Okay, maybe anyone else? If you have a question for Dr. Peter, you can raise your hand, please.
Yeah, maybe me, Budaya. Is it possible? Yeah, Dr. Catur is the colleagues of me from School of Pharmacy Microbiology in ITB. So I mean, is it hello? Is it clear?
Yes, Pak Edwin.
Okay. Yeah.
Yes.
Now my question to Dr. Peter. I'm interested on your about bioreactor, and then you show us if you have contaminant on the bioreactor, then you just put you have a diagram or a scheme how to reduce the contaminant. But in fact, yeah, there is a slide on that. Yeah, and
wait,
then
let me see. So like I said, the problem is that's not really part of my daily work. I just included it for you to get some okay. So that's also not like our company procedure or so. It's just to give you a general overview. What I can tell you for that is basically that at nearly every step of the production, so might it be inoculum, preparation of the media, loading of the fermenter, fermentation process, purification process. We have different sampling points where we pull samples for bioburden, for endotoxin, for mycoplasm, and so on and so forth. So, but it really depends on the product. So there's not like a routine way of doing it, but yeah.
Okay.
So like on every time it is checked, and it is then decided along the criteria which are given by our quality agreements and so on, whether we whether it's in good shape or whether you had to discard the batch basically.
So I mean, if you have lost, for example, 1,000 liter, and then you got contamination 5%, and then can you still reduce it, or you just to repeat it? No.
Yeah, no, most of the time you have to discard it. So that's really, that's really bad. And as you can imagine, I think the biggest fermenters we have are up to 20,000 liters. And so you run these fermentation campaigns for 7 to 14 days. And the later you lose your product, or the later you notice that you have a contamination, the worse it is, because it's like a huge financial loss if you have to flush your whole fermenter and get rid of the batch. So
that's why also, I mean, how about the waste management if you discard it?
Yeah, that's the next thing. So we have own waste management plant. Like you said, you cannot flush this just down into the sewers, because you have cells inside, you have bacteria inside, you have genetic manipulated organisms inside, of course.
That's right,
you might have antibiotics inside. And so it's given to the waste treatment plant, and they have basically like it's like a big commercial plant where they do the we have basically our facility, because this is a problem. Luckily, I can say that our controls are, I think, quite good, and there are not many runs that we actually have to discard, because, like I said, these hygiene procedures and robust processes are, I would say, quite elaborated. But yeah, that's just what I heard. So like, I'm not working with it directly in my day.
Okay, thanks a lot.
Yes,
Peter.
yes.
Yeah, nog dank. Yeah.
Okay, thank you, Pak Andri. Pak Edwin.
Yeah, there is still a raise hand from Bu Catur, ya, Budaya. There is a raise hand there.
Raise hand. Maybe Pak Arif already in front of his laptop. He got a good signal already. Pak Arif, can you hear me? Pak Arif? No.
No.
No,
okay.
I think not.
No. Okay.
There is a raise hand from Bu Catur. Is it alright?
Bu Catur. Okay. Yeah. Please, Bu Catur.
Okay, good afternoon. Thank you for the opportunity. Dr. Peter, I would like to ask you, is in your company you use a natural salt? Normally, natural salt.
Salt.
A lot of microbial contaminant. How do you handle it?
Excuse me,
sorry,
Bu Catur, can you repeat it again?
It was a little bit hard to understand.
So I think there is a.
There is a.
Can you just please?
Right.
I will write my.
Just ask again. Sorry.
Yeah, because the signal is very bad. Yeah.
Yes.
Yeah.
Yeah, it was hard to understand for me.
Yeah. Yeah, maybe Bu Catur can write it. Yeah. Okay. Okay, maybe the next question, Budaya, while waiting, Bu Catur writing, maybe.
Oh, okay, Pak Edwin. We have a next question here. Yes, Pak Edwin. Oh, no,
Yeah, not yet. Maybe for the audience, for the student, maybe.
Okay.
For the student.
I see here Edo Danielian will ask the questions.
Yeah.
Yes.
Actually, I would like to ask about the perspective of pharmaceutical industry towards the drug-resistant bacteria. And as we know that the mutation within the bacteria consortium is the thing that we can really control. And it poses to drug-resistant bacterial strain emergence within the consortium. And also the impact of drug-resistant bacteria is more serious in hospitalized patients, and particularly for vulnerable groups such as immunocompromised patients, such as the patient that receiving chemotherapy, having undergone organ processing, and things like that. And these patients are also exposed to high risk of colonization by resistant bacteria through the contact with healthcare delivery service. And my question is how the pharmaceutical industry see this problem and how the strategy in dealing with this problem, and what is the most challenging thing to create new drugs for specific resistant bacteria? Thank you.
Very, very good question. It's yeah. So me being a antimicrobial compound researcher myself and working a lot of resistance mechanisms and so on, it's a huge, huge topic. The problem is that the sales aspects of antibiotics are not so great. So if you compare it basically with cardiovascular drugs, which you would take for 10, 20, 30 years, pharmaceutical industry can sell a lot of these drugs. If you think about antibiotics, you might take them for 7 to 14 days, the worst case. And yeah, so the sales that you can do with antibiotics are not that high. I mean, that's the first thing why antibiotics per se are not so attractive for the pharmaceutical industry. The second thing is that if you come up with a new compound or a new antibiotic which has really good effects, most likely it will not hit the market directly, but it will be kept as reserve antibiotic. So it's basically the same. So you put a lot of money into the research and development, and then you're not allowed, or you cannot market it on a broad scale. So that's the big dilemma of antimicrobial research and production. I think there during the last 5 years, maybe there's a lot of more attention now going on. And also what I really like a lot, while Boehringer was, or my company was not focusing on anti-infectives, they have this vision that within 10 years, they also want to go back into that field. That's, I think, very important, because I think in Europe, there's only like one company left in Austria, which still produces antibiotics. And so maybe that's just to elaborate a little bit on the meaning of antibiotics in the pharmaceutical industry. And the second thing, if I read it or if I heard it correctly from your question, is what to do else. I think the time of broad spectrum antibiotics, basically where you give something which is very unspecific, which kills every bacteria, also the beneficial ones in your body, I think this time should be over. It's not over yet, but it should be over. And we really have to think about more specific ways to target the microbiome and to boost it, to target like only single bacterial species, for example, like phage therapy or coupling a phage endolysins to antibodies and so on and so forth. I think the research community or academia really hits nicely into this direction. Big pharma, not so much yet. It's more like they have some, they try to get in contact with startups or academic institutions and see whether they might get something out of it. But yeah, the problem is really marketing or selling antibiotics is, I think it's a difficult business for a normal pharma company. Yeah, I think public-private partnership would be important in that case. Sorry, long answer. I hope it's okay.
Thank you, sir.
Thank you. Okay. Yeah, it's really a complicated situation if we face this resistant bacteria in
Yeah.
the company. Yeah, right. Okay. We continue for the next question. Buchatur already have been right here. So the question is, do you use natural source for your product? If yes, how you handle it? Because normally they are source of microbial contaminants.
Correct. Good point.
Okay.
Natural products are important, but the contamination is always a problem with these. So also here the quality systems are in place, that basically the supplier where we buy these natural products from are qualified, so that we know they have in-house testing, they have in-house quality checks, that the product they deliver is good and the microbial counts are low. But we also check these ingredients before we use it in our plant. And also here you have to differentiate a little bit, because, for example, for biopharmaceuticals, which you would inject or which are sterile products, I'm not sure whether a lot of natural products are used because of this problematic. In our case, for example, we are using carnauba wax, which is from the carnauba tree, carnauba palm tree, and this is also a natural product. But since it's only orally taken and the process is running at high temperatures, we don't have this big problem with contamination. But for sure, natural products from natural sources are a big problem. So you really have to handle this within your quality management systems. Yeah.
Okay. So this is how do you okay, you handle that natural source. Okay.
Yeah.
Okay. Now we go to the next question here from Laura Yola. I would like to ask a question: how do you ensure that every effort towards a sterile production area and proper quality control is not doing any harm to the environment?
Good question. Big topic nowadays, and difficult topic for sterile production, especially because with some points you don't have too many alternatives. Thinking about sustainability, for example, especially not in sterile production. So there's a lot of plastic use, lots of chemicals, and so on. And maybe I go back a little bit in the German chemical history. So just Germany was always a quite big player in chemical and drug production, and they had back in the times low standards. So lots of the old production plants are quite what do you call it? There's a lot of chemical waste lying there. And this was bad, but that also led to the fact that lots of initiatives had to be brought up, lots of law had to be brought up. And for example, we have a huge EHS department. This is environmental health and safety. So these are engineers which basically check all the chemicals, check all the stuff that we are using. They check the processes, and they also check the disposal. Like in the first question of Edwin, like what do we do if a batch goes wrong? It's put into this waste treatment plant, and they see that it's basically worked up accordingly, so that there's no harm for the environment. Also, I'm not a specialist on this, but I have a friend working in the EHS department, and I think they are very well prepared. You always hear it. You can Google it. There's some occurrences, or in Switzerland from Novartis, where they basically put lots of toxic chemicals into the Rhine River. And unfortunately, my company also was involved in an accident in the, I think, 1950s or so. And I think also like the huge API production facilities in Pakistan and India, they have lots of problems. So they produce high quality product, but environmental standards are not that high. And I think that's good in Germany, because the government was very active about that. So yeah, I think it's rather elaborated here. So, but yeah, it's sterile production of chemicals and pharmaceuticals is always difficult. You always have an impact on the environment, and that's what makes it also very, very expensive for the companies to treat the waste. Yeah.
Okay, so it should be already like a waste management system already.
Yep.
Okay. Okay. There are questions from Nadifa. Can you speak up, Nadifa Putri? Can you speak up?
Yes. Yeah. Thank you for the opportunity. So my name is Nadifa, and as you have mentioned before that based on the risk priority number, the room in your company, the room with low RPN is monitored quarterly, the medium RPN is monthly, and the room with high RPN is monitored weekly. So what I want to ask is there any chances, or did it ever happen in your company that the monitoring period of these rooms is changed due to some problems? Like, for instance, the room with high RPN is monitored twice a week and so on. Thank you.
Yeah, definitely. Also, these are only the values for our production site. Doing like non-sterile drugs. For sterile, even the monitoring is higher. But yes, and it's also very, very important prerequisite, which is demanded by the authorities, that this risk analysis is reworked from time to time. So maybe every year, every half a year. So that's what we do. Like there's a revision going on. And also if you have special incidents, for example, we had one storage room where we had spore formers coming up again, and we couldn't get rid of them. So there we really put the monitoring frequency higher, that we could keep track of it, because otherwise you just don't stand any chance to see where this contamination really comes from. And that's also the important point, that you do this investigation, so where the contamination might come from, with a scientific approach. Yeah, it's always just a problem, because it's always bad for the production if you have contamination in your rooms. But for me as compliance officer, it's always a little bit more fun task to do, because you can investigate, you can think, where do the bacteria come from, what kind of bacterium is it? And yes, also, so short answer, yes, we do that definitely, and we have to do it. And it's also demanded by the authorities.
Okay, thank you very much.
Okay, is it clear? Okay, now the next question is from Aisha. You said before in the past answer that in your company was using the recombination or imitation microbes to recycling the waste product for produce the antibody based on your normally use. What the usual imitation bacteria that you use to recycling the waste from antibody production?
Okay, wait, I have to read it again.
Yes.
Okay, I get the question. So yeah, in the waste treatment plants, you have specific bacterial cultures, but it doesn't matter whether it's a pharmaceutical company or not, or it's just your public sewage system. They all more or less have, I think, the same bacteria which they are using. And for inactivation of our antibody production, so for inactivation of cells, but also for antibody leftovers, because you might get product or the wrong antibody into the next fermentation process. So we have validated cleaning procedures for that. For example, treatment with acids and bases. So specific cleaning agents which are proved to reduce or to destroy the biopharmaceutical and the bacteria and so on. And this is always checked. So this is also a big part of our quality management system. I hope that answered the question. What the usual imitation bacteria did you use? So I cannot tell you what the bacteria strains are which are used in the waste plant, because I'm sorry, I'm not an expert on this. Yeah.
Okay, no problem.
Maybe it's a secret of the company, right?
Definitely. No, I think this is actually a very specific topic, because depending on what kind of waste goes into the sewer, you would need different cultures to work up the chemicals and the leftovers. But sorry, I don't know that.
Okay, thank you, Mr.
Okay, Pak Arif,
Yes.
have you been there? Okay, now go
Yes.
back to Pak Arif. Okay.
Okay, I'm taking over the role as a moderator. Is there any other question for Dr. Popella? If not, maybe I have some question. I don't know whether it's already asked or not. Yes. Other questions? Okay, no. So in this kind of pandemic, as you presented that the management systems, like the protocol, the procedures in the boarding Ingelheim is so sophisticated and well established. Do you have any adjustment or change that you need to do in the case of the pandemic? Because the system that you have already in the boarding is already very hygiene, kind of like that. And the second answer from me is Boehringer Ingelheim also participating in the discovering or developing the vaccine of the coronavirus? Yeah.
I start with the second question first. They are not involved in the development of a vaccine, and at least what I know. What we have going on right now is it was at least in the local news. We have an antibody against COVID, which was produced in our clinical production plant. So basically, we have clinical trials going on with this antibody. And this antibody is, we have a respirator platform. So like it's like an asthma spray where you inhale. And I think it's, they aim at giving this antibody by this respiratory system directly into the lung. So if you were exposed to the coronavirus, that you basically have this to neutralize it. That's what they are doing. Vaccine, I don't think so. It's also not something which the company is very strong. So they're doing animal vaccines, but human vaccines, I think we have an HIV vaccine. No, not vaccine, but some prevention drugs, but no, not that I know at least. And for the first question, regarding our production processes, there wasn't a change, at least not that I know, because I'm also sitting in the clean room, and for me, it's always the safest place to be because everything is clean. That's quite nice. But we had lots of activities in our offices. So I think most of the people are in home office right now. You have to wear masks. They disinfected the tables. They're trying to really separate the working shifts. So we have three working shifts, especially in the biopharmaceutical production. So they're producing 24/7. There are always people, and if you imagine there's one infected, then it's really hard to. What should you do with a shift? Basically, you would have to send them all home, and the production process would stop. So that was not so much from the production or from the production process challenge, but from the pure economic and economic challenge and organizational challenge, but not so much during the production process, because this is, like you said, it's very elaborated, and the clean room is the safest place to be.
Yeah. Thank you very much.
Yeah.
There is also another question from Dr. Riani. Dr. Riani, would you like to directly ask Dr. Popella, or should I read the chat? Maybe I will just read the chat.
I can read it also.
Okay. Yeah.
You have data based on contamination. Good question. Yeah, that's actually one of the more interesting microbiology topics in in the industry, at least for me. Unfortunately, I since I'm not in the department who's doing this identification, I don't know too much about it. But yes, so one thing that is also demanded by the authorities is that you do a regularly identification of the microbes found in your production facility, and these are basically labeled as household microbes. So which you would typically find. So every production facility has its own set of microbes. It's just how it also is. It's like a fingerprint basically. And I think from time to time they get new bacteria. They find new ones, at least to the species level. So they're doing. I heard they're doing a lot of multi-TOF identification and 16S RNA sequencing, and from time to time they find something. But I don't think that these are like really and never identified bacteria because these are just all routine processes, and so on. And that's for me always, it's a little bit disappointing because like I told you the story of the spore forming bacterium. It was one which I'd never heard about it, and then I researched a little bit. Oh wow, this is interesting. Do you ask the guys in the microbiology department, do you know anything about it? We don't care as long as it just has to be gone. So, okay, it's just the academic spirit that's always in your head. And yeah, how you identify or make confirmation of them? Yes, like I said, MALDI-TOF and 16S RNA sequencing and tracing source of contamination in your production process. That's actually the very, very, very hard part because most of the time you do these root cause investigations, and yeah, it's difficult. There are many, many factors involved. For example, like if you have a contamination, then you do sampling, then it takes time that the bacteria grow, then it takes time to identify themselves, then it's maybe already too late, the sterilization process has already gone through equipment and rooms, and yeah, that's, I think finding this is a huge challenge. And I know because a friend of mine is working on that, is that we have an own project to push that even further. I think so far we never had a problem with this, but it's really a huge challenge. I think most of the time it's like I said, it's human intervention, at least in the biopharmaceutical production process, which most of the time is a source for contamination. Yeah. But it's hard to trace actually. Yeah.
Okay, thank you for the answer. Is there any other questions? I think no. So as you said that before, that you found so many, not many, maybe some contamination in the production process. So is it not really, or you don't want to use any antibiotics in the reactor or in the fermentation process, right?
Yeah,
what is
right.
the cause?
Well, why not? So one thing which is which one is not aware of if you're not working in the pharmaceutical industry is that the recipes that you're using for production are very limited. So they're all registered. Basically, you're not allowed to change anything. You are allowed, but it has lots of hurdles if you want to change anything. So basically, we're running a product for 10 years, and at one point say, maybe why are we not adding an antibiotic that would maybe kill or reduce biocontamination? It's just not possible because the regulatory authorities are very strict on that. Economic cost is a big factor because you would have to add a lot of antibiotics in a 20,000 liter scale fermenter, and also it's hard. So producing these large biopharmaceuticals is actually very difficult because you have to take glycosylation of the compound into consideration, inclusion bodies, agglomeration, precipitation, and so on. And you never know how your eukaryotic cells might react on antibiotics because they will also be a stress for them, and maybe they might produce a faulty product and you wouldn't even notice it. So like I said, I'm not a specialist for this. I guess there are some processes which involve some kind of antibiotics, but not in the big fermenters at least. Yeah. That's why the hygiene is so important. Yeah.
Thank you, Peter. Is there any other question?
Arif, can I ask one question, but out of topic maybe?
Yes, please.
Yeah, Peter, once again, this is maybe out of topic with the research, but topic with drug competition in Germany. because I heard Bayer is the biggest for the pharmaceutical industry, and then how the Boehringer Ingelheim compete with Bayer, etc. So you know the scale of that, and then is it any competitor with Chinese medicine or something like that in Germany?
Yeah, so as I said, Germany was always had a very, very strong pharmaceutical production industry. It now shifted a lot. It's still shifting, especially API production to Pakistan and India, and also China, of course. China is on the rise. They're producing lots and lots. And yeah, Bayer, of course, is a competition for us. We have similar products. Yeah, for example, we have Bayer has Xarelto, which is really, I think that's on the one slide here. It's really it's a very, very well selling drug. Let me see. I think it should be here, Xarelto, yeah. And he has a 6.8 billion of sales. And we have a similar drug, Pradaxa, which is in many in the opinion of the company, it's superior to Xarelto, of course, but Xarelto is just selling better. And this is, as I'm saying this a little bit unofficially, but Boehringer is, since it's not listed on the stock market and it's still family owned, it's a little bit more conservative, maybe a little bit more careful. They're not so strong in market penetration. We more focus on good manufacturing and this contract manufacturing. So Bayer is very, very well known in Germany. Everyone knows Bayer, 50,000 employees. Boehringer Ingelheim, 50,000 employees. Not so many people know it. But that also stems from the thing that Bayer produces a lot of over-the-counter drugs, so which you can just go to the pharmacy and buy. And we produce a lot of drugs which you would just get in the hospital. So it's just doesn't make sense. Oh, yeah, they just don't know it. And yeah, China is so I think most of the big pharma companies have ties to China now, because you're not allowed to produce in China as a foreign company without building a partnership. And so it's
fake medicine also from China.
yeah. So China is a huge challenge actually, and but we're active there. It's a big market. It's an important market, and I think it will be more and more and more. Yeah, definitely.
Okay, thanks, Peter.
Yes, you're welcome.
Yeah, Pak Arif, thank you.
Thank you, Pak Edwin. Is there any other questions? I think there's no other question. So thank you, Peter, for your very nice talk. Very good talk.
Good. I hope I didn't run you over with the car. I hope it was not too much information. I hope it was at least a little bit interesting. Maybe I can close with 2-3 sentences. So
yes, please.
From my experience, it's not so easy to get into the pharmaceutical industry as a microbiologist, at least not in Germany, because you have this pharmacy studies and so on. There are a lot of people. But as soon as you enter, they're also, you're a microbiologist, that's great, we need those. I was like, okay, I didn't notice that until now. And yeah, I think it's a good field of industry. Now with COVID, many of my friends they cannot work or because they're closing, and pharmaceutical industry just runs and runs and runs. You're actually really helping people with your products. I think that's great. And if you're interested to go into pharmaceutical industry, I would say that try to get an internship as early as possible. Read about pharmaceutical industry, about regulations, because it's very, very different from what we have at the university. That's just the advice that I can give you. So reach out to people, listen to talks, and maybe get an internship. So unfortunately, like some work is boring. It's not as thrilling as academia, but they also pay well. So.
That's the main point.
No, but it's, I can definitely say go to pharmaceutical industry, and it's a good place to work actually.
Okay, yeah,
And
thank you.
if you have any more questions, you can get in contact with me. I'm on LinkedIn. You can add me there, or you can write Arif, and he can give you my email address, and so I'd be happy to
Yes.
help in any case.
Okay, very nice. Yeah, I think there is no other question, so we will just close this discussion and talk. Once again, we really appreciate your time, Dr. Peter Popela. Very nice talk, very nice discussion. And also thank you for the audience. Yeah, thank you very
Yes.
much. See you next time. So Peter, if you have any chance or possibility to come to Boehringer Ingelheim branch in Bogor, Indonesia.
In Bogor, yeah. I actually I asked whether I could go there for a rotation, but they said no.
You will be shocked
Try
if you
again, and then you can come to us.
Indonesia. Yeah.
Indonesia, Bogor. Okay.
Yeah.
Bogor, yeah, right. We have a plant there, but no chance for me to go there until now.
Just go here for vacation. It's also fine.
Yeah.
I think that's a very good plan. As soon as the pandemic is over, I will come to Indonesia.
Okay, yeah. Thank you very much.
Thank you very much.
Yes,
No,
you're welcome.
thank you.
You all
Next time.
stay safe and healthy. Watch out for yourselves, and see you around. Bye bye.
Thanks a lot.
Bye.
Bye.
Thanks a lot. Bye bye.
Thanks a lot.